Hi friends, today we will revise from Pathfinder book, and the first topic we are going to study is molecular genetics. So in this book there is molecular genetics, biotechnology, plant physiology, human physiology, plants and fungi, animals, ecology, evolution. So let's start with molecular genetics first. So first of all, what is genome organization? In the first heading, genome organization, where genome is defined as the totality of all DNA of an organism. It's a totality. And genomes, prokaryote and eukaryote, they are made up of DNA, but few viruses have RNA genome. And this eukaryotic genome consists of two distinct parts, nuclear genome and organelle genome. Organelle genome is mitochondrial chloroplast. Okay, and the amount of DNA present in the haploid genome, okay, amount of DNA in the haploid genome is the C value. C value is the characteristics of each species. C value is the characteristics of each species have a different C value. The value ranges from around less than 10 to the power 6 base pair as the smallest prokaryote, mycoplasma to have more than 10 to the power 11 base pair for amphibians. Okay, so the genomes of higher organism contain a large excess of DNA. Okay, as the organism evolves, the C value increases, DNA increases. Okay, so DNA content of the haploid genome is related to the morphological complexity of the lower eukaryotes. So DNA content of the haploid genome, it is related to morphological complexity of lower eukaryotes, but this varies extensively among higher eukaryotes. Okay, so haploid content is, it's a complexity of it's related to the complexity of lower eukaryotes, but there is variation in higher eukaryotes. So in this picture you can see that the DNA content of the haploid genome of a range of phyla, the range of values within the phylum is indicated by the shaded area. Okay, the range of values within the phylum, okay, the values, if it's more, the shading is more. Okay, now, so the genome of different species of amphibians can vary from 100-fold in their DNA content. Okay, so genome size in some eukaryotes, it's different in different organisms. In yeast it is 12 genome size. In Arabidopsis thaliana genome size is 120 megabase pair. In Drosophila melanogaster it is 170 megabase pair. In Homo sapiens it is 3300 megabase pair. In Hordeum vulgare barley it is 5300 base pair. Next is, what is genome complexity? So genome complexity is the total length of different sequences of the DNA. First of all, what we studied what is genome. Genome is the totality of the DNA. What is genome complexity? Is the total length of different sequences. Okay, total length is the complexity. So this genome complexity can be measured by renaturation kinetics of the denatured DNA. Okay, how easily it can renature, it determines genome complexity. So renaturation of DNA, this occurs through complementary base pairing. Renaturation of DNA depends on random collision of the complementary strands and follows second order kinetics. So renaturation follows what? It follows second order kinetics. Okay, and it depends upon random collision of the complementary strands. So a DNA association reaction, it is described by cot half. Okay, DNA association reaction is described by cot half. If large DNA is sheared into uniform fragments and they are allowed to renature, then the rate of renaturation of the denatured DNA is expressed as dC upon dt is equals to minus kc square. So this genome complexity, it is measured by a formula, dC upon dt is equals to minus k c square. So what is k here? It is the second order rate constant. Capital C is the concentration of single stranded DNA at time t, and the second order rate equation of the two complementary strands coming close together is given by the rate of decrease in C. Okay, when they come close together, it is measured by the rate of decrease in C. So starting with a concentration of C0, where the DNA is denatured at time t0, where it has not renatured yet, the amount of single stranded DNA remaining at some time t is, so single stranded DNA remaining is final by initial concentration is equals to 1 by 1 plus K. What is K here? K is the second order rate constant. Okay, second order rate reaction is minus Kc square. K is the constant and C is the concentration. Okay, and what is the what is okay, so the product of, so what we found, that C by C0, which is final by initial concentration, is equals to 1 by 1 plus K C0T. So the time for half of the DNA to renature, that means C by C0 should be 0.5, and it is defined as T is equals to T half. So when when time for half of the DNA to renature is 0.5, just putting the value of C by C0 0.5, so is equals to 1 plus Kc0 T half is equals to 2. When you will solve this, you will find that this is equals to 2. So yielding what? Cot 0 T half is 1 by K. Yielding what? Cot 0 1 by cot 0 C0 T half is 1 by K. So the product of C0, the product of C0 T half, it is called as cot T half or cot curve, okay, which is 1 by K, which is inversely proportional to the rate constant. So since C0 T half, it is the product of the concentration and time, it is the product of the concentration and time. Cot curve is what? It is the product of concentration, which is C, and time required to proceed halfway. A greater cot implies a slower reaction. So when the as fast as as more the value of the K, more faster will be the reaction. And when it is reverse, okay, when it is reverse, when there is lesser value of K, okay, when the K will be higher, the cot is inversely proportional, cot will be lower. Okay, so a greater cot half implies a slower reaction. Why? Because K is the indication of faster reaction, and if K is higher, the cot value will be lower. So the reassociation of DNA, it is followed in the form of cot curve, and the graph of the fraction of single stranded DNA re-annealed C by C0 as a function of cot on the semi logarithmic plot is referred to as cot curve. So what is basically cot curve? Cot curve is a graph of the fraction of single stranded DNA re-annealed as a function of C0T on a semi logarithmic plot. Okay, so you can see here, this is a cot curve of double stranded DNA from the indicated source. Okay, so what it is basically, it is the graph of the fraction of single stranded DNA, okay, which is re-annealed as a function of concentration into time on a semi logarithmic plot. So fraction is reassociated is half, which is marked here. 100% reassociation is at the top, and starting to just reassociate is at the lower side. So the rate of the reassociation, it can be followed spectrophotometrically by the UV absorbance decrease as duplex DNA is formed. Okay, when there is duplex DNA, then there will be low absorption, low reading. So a cot curve describes the kinetics of reassociation of a sample of a double stranded DNA that has been fragmented into smaller pieces, denatured to single strands, and then permitted to reannul. So DNA taken, fragmented, and permitted to reannul. The cot half of the kinetics reactions, this indicates the total length of the different sequences that are present. So what does cot half indicate? It indicates the total length of different sequences that are present. So this is kinetic complexity, and the cot half for the renaturation of the DNA of any genome, it is proportional to its complexity. Cot half, it's proportional to its complexity. More the cot half, more is the complexity. So complexity of any DNA can be determined by comparing its cot half with that of standard DNA of known complexity. Okay, so you can compare the complexity of the DNA by comparing its cot half. Okay, the more the cot half, more the complexity. So question is there, repetitive DNA sequences were first identified by studies of rates of DNA reassociation. What relative rate of reassociation are expected for sequences repeated 1000 times in the genome compared to genes with only a single copy? How will you compare? One gene is repeated 1000 times, one gene having single copy, where one gene is in single copy in the genome. How will you compare? You will compare with DNA reassociation study, which is a biomolecular reaction. So the reaction rate is proportional to the square of the DNA concentration. You can see the picture, the formula. It is Kc square. So the reaction rate dC upon dt is equals to minus Kc square, which is the square of the concentration. Okay, so a sequence repeated 1000 times in the genome would therefore reassociate with the rate of 10 to the power 6 fold, which is greater than that of single copy of the DNA. So more larger the DNA, larger will be the reassociation, and this is how you calculate, because complexity, the rate of the reaction is proportional to the square of the DNA concentration. More the DNA concentration, the rate of reassociation will also be more. So next we come to repetitive sequences. What are repetitive sequences? So reassociation kinetics is what we are studying. It identifies two general types of genomic sequences, non-repetitive and repetitive. So non-repetitive means they are unique and there is only one copy in the haploid genome. But what are repetitive DNA sequences? That they are present in more than one copy in the DNA sequence. And the proportion of non-repetitive and repetitive DNA sequences, they varies widely. Prokaryotes contain only non-repetitive DNA. Please remember, prokaryotes contain only non-repetitive DNA. Similarly, in lower eukaryotes, most of the DNA is non-repetitive. So lower eukaryote and prokaryote, they have non-repetitive DNA. And in case of plants and amphibians, repetitive components may account for up to 80% of the genome. So in general, the length of the repetitive DNA component tends to increase with genome size. More the genome size, more the repetitive DNA. Okay, more the reassociation when the complexity of the genome or when the concentration of the DNA is more. So renaturation kinetics further divide the repetitive sequences into two general classes. So first of all, one is non-repetitive, another is repetitive. Repetitive into two, moderately and highly repetitive. So highly repetitive DNA sequences are what? Highly repetitive DNA consists of very short sequences repeated typically 100 base pair they are less than 100 base pair. They are repeated many times in tandem large clusters. Okay, they form a cluster. So because of its short repeating unit, it is sometimes known as simple sequence DNA. Because the repeat is short and it is simple sequence DNA, such simple sequence DNA can often be separated from the bulk of the chromosomal DNA. It's easier to separate them by shear degradation followed by density gradient ultracentrifugation in cesium chloride. Okay, because they are short, small, they can be easily separated by CSCl. Equilibrium density gradient ultracentrifugation, that constitutes most common method for DNA separation. Okay, how do you separate the DNA? It's using CSCl method. So it separates the DNA on the basis of their differences in Bowen density. CSCL uses the methodology of Boolean density separation, and this Boolean density row of double-stranded DNA depends upon its base composition. Okay, the Boolean density row of double-stranded DNA depends on its base composition. Rho is equals to 1.660 plus 0.00098% GC gram per centimeter cube. So there are distinctive based compositions of simple sequence DNA, and their difference in Boolean density, it results in the formation of satellite to the main DNA band. Okay, so since there is a difference in Boolean density, and this result in the formation of satellite, the sequence of these DNA is known as satellite DNA. So how to understand about satellite DNA? For example, when mouse DNA is centrifuged on the density gradient, two types of band will be distinguished: main band and satellite band. The main band will be 92% of the genome, and it's centered on the Boolean density of 1.7, and there is a smaller peak which represents 8% of the genome. There's a distinct Boolean density of 1.69, which is a little lesser than the main band. Okay, so you can see the graph Boolean density. Most mouse DNA is separated into the main band and the satellite band by centrifugation through the density gradient CSCL, and you can see that the satellite band have Boolean density lesser, quite less than the main band. So satellites are present in many eukaryotic genomes. They may be either heavier or lighter than the main band. Most of the time it is lighter. And one type of human satellite DNA, the alphoid DNA, it repeats found in the centromeric region of the chromosomes. So one type of human satellite DNA, which is alphoid DNA repeats found in the centromeric region of the chromosome. Okay, so example of human satellite DNA, if it's asked, it is alphoid DNA, which under repeat of alphoid DNA, it's found in the centromeric region of the chromosome. Human genomic DNA forms a main band. Human genomic DNA forms the main band, which has the Boolean density of 1.701, and the three satellite bands at 1.687, 69 and 697. So these are like all have smaller Boolean density than the main band. And the satellite DNAs, they are species specific. A single genome can contain several different types of satellite DNA, each with different repeat unit. So these units being anything from less than 5 to 200 base pair, greater than 200 base pair, and satellite DNA sequences, they are generally not transcribed and are located in the heterochromatin associated with the centromeric region. So what we understood about satellite, they are having a lesser Boolean density, they are species specific, and what else, that their sequence is known as satellite DNA. Okay, and they are less in the genome, and example is alphoid DNA, which is present in the centromeric region of the chromosome, and they can be less than 5 to greater than 200 base pair. They can be different types of satellite DNA, each have different repeating unit, and they are not transcribed into RNA, and they are located often in the heterochromatin associated with centromeric regions of chromosome. So mini satellite and microsatellite. Mini satellites and microsatellites, they are tandemly repeated DNA. Okay, mini satellite and microsatellite, they are tandemly repeated DNA, and they are classed as satellite DNA. They are tandem. Okay, but they are not appearing in satellite bands on density gradients. So you can see the density gradient of the genomic DNA and the satellite DNA, but you cannot separate mini and microsatellite based on the density gradient. Mini satellite form clusters up to 20 kb in length with 20 to 50 repeat units, and each containing about 15 to 100 base pairs. Okay, mini satellites form clusters. It will form cluster for about 20 kb in length with 20 to 50 repeating units, okay, with 20 to 50 repeat units, each containing about 15 to 100 base pairs. Mini satellites have 20 kb up to 20 kb in length, and each consists of 20 to 50 repeat units. Okay, remember, it consists of 20 to 50 repeat units, and the number of bases which it contains, it is around 15 to 100 base pair. Next is telomeric DNA, which in human comprises hundreds of copies of the motif. Now it is a very common question, what is the sequence of telomeric DNA? It is TTA GGG 5 prime to 3 prime. It is an example of mini satellite. Okay, microsatellite. So mini and micro, difference you learned. Microsatellite clusters are shorter. Mini is bigger, micro is even smaller. They are shorter, usually less than 150 base pair. Microsatellite, they are less than 150 base pair, and the repeat unit is usually 13 base pair or less. Okay, so micro have the unit less than 13 base pair. Mini have unit up to 20 kb. Okay, so the typical microsatellite consists of how many repeats? It consists of either 1, 2, 3 or 4 base pair unit repeated 10 to 20 times. Okay, mini satellite are repeated even more, they are bigger, and they have 20 to 50 repeat units. Each repeat unit can be 50 to 100 base pair. Okay, so but here the each repeating unit is what, 10 to 1 to 2 to 3 to 4 base pair unit. Okay, so less than 150 base pair. So difference is very important. So satellite DNA, what is the size of satellite DNA? It is 5 to 171 repeat. So when we will discuss this comparison, then it will be very easier for you to remember to like compare. So satellite DNA, we have understood that they are different, like they are around 8% in the genomic DNA, and alphoid is an example, and have lesser density gradient in CSCL than genomic DNA, and they are mainly present in the centromere, having 5 to 171 size of the repeat unit. Then it's alpha satellite, which has repeat unit size of 171, very big. Then beta satellite, it is 68, beta is smaller. So alpha satellite is generally at the centromeric heterochromatin. Beta satellite is the centromeric heterochromatin of some chromosome, limited availability. Mini satellite are bigger, 9 to 64 base pair repeat. They are closer to telomere. And micro satellite, they are 12 base pair and they are dispersed throughout. Now the question comes, you are given two unlabeled tubes, one containing human DNA and the other containing DNA of a bacterial plasmid. One is human, one is bacterial. Now you have to distinguish these DNAs by reassociation kinetics. How you will do that? So it's very, you can say that the human DNA would be complex than plasmid DNA, but the curve would be similar. Okay, the curve for plasmid would be similar to that of observed E. coli DNA because they have single copy. Okay, the human curve will have multiple slopes which represent different classes of DNA. So single copy will show single curve, and you will see multiple slopes when you will see different classes of the DNA that reassociate at different rate. Okay, so based on reassociation kinetics, you can distinguish in that way. So that was highly repetitive. Highly repetitive, what we studied about satellite. Okay, highly repetitive, if we revise what we studied in highly repetitive, we studied what? We studied about satellites. Okay, so in repetitive structure, it is moderate or high repetitive. In high repetitive, we studied about satellites. Next is moderately repeated sequences. So they consist of short sequences that are typically 10 to 1000 times in the genome. They are quite like you can say 10 to 1000 times, and they are dispersed throughout. Okay, so you can see highly repetitive are generally in the centromere or heterochromatin, but these moderately repetitive, they are dispersed throughout like microsatellite. So a significant fraction of the moderately repeated DNA sequences of mobile DNA elements, short sequences of the DNA that have the ability to move to the new location in the genome to make additional copy of themselves. They are like transposable elements. So they move very rarely, but transposons, which are considered as selfish DNA, and these are defined as sequences that do not contribute to the genotype of the organism but propagate within the genome. Okay, they propagate within the genome. Next, if we study deeper about these mobile genetic elements like transposon, which is very necessary to understand. So mobile genetic elements, they are moderately repeated DNA. Highly repetitive are satellite. Mobile genetic elements are moderately repeated DNA sequence, and they are interspersed at multiple sites throughout the genome. Okay, they are interspersed at multiple sites throughout the genome. So genome generally contain many varieties of transposable elements. Mobile DNA elements encode enzymes that can insert their sequences in new sites in the genomic DNA. They can copy paste or they can cut paste themselves in the new position, and this is known as transposition. So there are two kinds of transposable elements. There are two kinds of mobile DNA elements, transposons and retrotransposons. And transposable elements that transpose to new sites directly as DNA are called transposons. So some move by conservative process, which involves the excision of the sequence from original position followed by reinsertion, like cut and paste mechanism. Okay, so number of copy does not increase in this case in cut and paste condition, which is a conservative process. Okay, the conservative people, like, they do not change. In that way the copy number will not increase. Okay, second type of transposition is called replicative transposition, where there is increase in copy number. Replication will occur, number will increase, copy number will increase. In this process, the original transposons remain in a place while a copy is inserted at the new position. So you can see in the picture, one is conservative, where it is removed from one position, added to another. Replicative is a new copy is added without disturbing the previous one. So transposable elements that are first transcribed into RNA of the element, which is then reverse transcribed into DNA, they are called retrotransposons. So retrotransposons, where RNA are converted into DNA and then they do the transposition work. So mobile elements are of two types, transposons, which moves by a DNA intermediate, and retrotransposons, which move, or there is a RNA intermediate molecule which is formed, which is then reverse transcribed into double stranded DNA. In both cases, the double stranded DNA intermediate, it is integrated to the target site of the DNA to complete the movement. DNA is the one which will move. So it can be by cut paste mechanism or it can by copy paste mechanism. So you can see the picture, whether it be transposon, whether it be retrotransposon, it's a DNA intermediate which will move, and thus it is transposing. Next is bacterial mobile elements. So bacterial mobile elements, so most mobile elements in bacteria, they transpose directly as DNA. But in eukaryotes, it's retrotransposon, which is more common. So both types generally produce what? They produce short direct repeats at the site of insertion, which flank the mobile element. Okay. So both types generally produce short direct repeats. They produce short direct repeats, okay, at the site of insertion, which flank the mobile element. Okay. So there are some repeats which is flanking. You can see in the picture there are some repeat which is flanking the mobile element, and the length of the repeat, it depends upon the type of mobile element, which we will discuss. So insertion sequences, they are the most common transposons present in bacterial genome. They are the insertion sequences, which is in short said to be as IS elements. And the general structure of IS elements, it is diagrammed in this figure. An inverted repeat usually contains, inverted repeat kind of, you can say, IS element. It contains about 50 base pairs present at IS element flanking is the inverted repeat and direct repeat. So inverted repeat is around 50 base pair, which you can see here. Inverted repeat is around 50 base pair, okay, and is present as at each end of the insertion sequence. Between the inverted repeats, it's a protein coding region, okay. Between the inverted repeats, it's a protein coding region, which encodes for one or two kinds of enzymes for transposition of IS element to a new site. And there is important hallmark is a presence of short direct repeat, okay, which is 5 to 11 base pair. Short direct repeat, which is 5 to 11 base pair, okay, which is adjacent to the both ends of the inserted elements. So it is towards the end of the insertion element. So one is inverted repeat, one is direct repeat, and here is the protein coding region. And the enzymes which is involved here is transposase. So this is a general structure of bacterial IS element. You can see that it is flanked by an inverted repeat at each end. The sequences of the inverted repeats are nearly identical, but they are oriented in opposite direction. So here it is written for only one. You can see two bigger arrows. This and this. These are inverted repeats, and they are nearly identical, and they are in opposite directions, okay. And this is the characteristics of IS element. And then there is a short direct repeat, which is 5 to 11 base pair in length, and this direct repeats become duplicated with one copy at each end. Here it should be also there, direct repeat, okay. And the length of the direct repeat is 5 to 11 base pair. So this was about the IS element. Second is composite transposon. So what is composite transposon? Composite ones are larger than IS elements and contain one or more protein coding genes, okay. Composite ones contain one or more protein coding genes in addition to those required for transposition, okay. So whatever is required in number 1 case, it is having some protein coding genes as well. So it is containing one or more protein coding genes. Say for example TN9. So this have the bracketing IS element, which you can see, and in between there is a protein coding gene, okay. So you can see that in TN9 it has a bracketing IS element in the same direction, whereas others TN5 and TN10, they have IS element in opposite direction, okay. It's not clearly shown in the picture. So TN5 have IS element in the same orientation, other have in different orientation. So composite transposons are what? They have, in addition to whatever is there in the transposon, it has some additional protein coding gene. So they are not the ones to carry resistant genes, like protein coding gene, you can say resistant gene. Such genes can also be So composite transposons are the one which can carry resistant genes. So they are not the only ones which carry resistant genes. There are also some other genes which carry resistant genes. They are non-composite transposons. So they are bracketed by short inverted repeats. They also have short inverted repeats, but the resistant gene is a part of minimum transposable unit. TN3 and TN21 are the example of non-composite transposon. So non-composite transposon, what they are, they can also be an integral part of transposon, but they are bracketed with short inverted repeats. Their inverted repeat is short, and the resistant gene is a part of minimum transposable unit. Okay, here resistant gene is a part of kind of longer transposable unit. Here TN, it is a part of minimum transposable unit. So in the next video we will cover bacteriophage elements and then eukaryotic transposons.